Hello, my name is Caroline Johnson, and I would like to first thank the judges for choosing me as a finalist from such an esteemed list of nominees. I recently joined Yale School of Public Health as assistant professor and was previously staff scientist in Gary Shuzak's lab at the Scripps Center for Metabolomics and Mass Spectrometry. My research career has focused on developing analytical and computational methods for the discovery of biologically important metabolites to enable a deeper understanding of the metabolic response to environmental exposures. To do this in the most effective manner, I've been lucky enough to be involved in the design and implementation of two widely used bioinformatic resources, XEMS Online, a cloud-based data processing software that has over 11,000 users, and the Metlin database, which houses over 240,000 metabolites. My contributions to the development of these platforms have enabled complex experimental analysis and consequently have fed back to tailor my experiments in the lab, helping to recover the broadest range of metabolites and remove interfering signals. In the next 5 minutes, I would like to present a short background on some of the issues associated with metabolite analysis and how my research has helped to alleviate them. Analysis of the metabolome is complex, as metabolites can arise from a variety of sources. In addition to being produced directly by the host, metabolites can derive from microorganisms, xenobiotics, and environmental sources. Metabolites play crucial roles in biological pathways, and their differential regulation can reveal novel insights into disease and environmental influences. Therefore, analysis of the metabolome must be sensitive, robust, and reproducible in order to accurately link metabolites to phenotype. The main bioanalytical tools I've used for metabolite recovery and identification in my research are untargeted and targeted mass spectrometry-based metabolomics. Untargeted metabolomics aims to measure the broadest range of metabolites present in an extracted sample without previous knowledge of the metabolome. These types of metabolites recovered are influenced by the extraction and an analytical method of choice, but result in a complex data set that require computational tools to identify and correlate metabolites between samples. Targeted metabolomics provides higher sensitivity and selectivity where methods are used and developed to optimize for the analysis of specific metabolites. I have used this tool throughout my research to validate my untargeted data and further explore specific metabolic pathways of interest, providing quantitative information. So my overall goals in my research have been to improve both the bioanalytical and data processing aspects of metabolomics in ways that enhance metabolite identification and strengthen statistical and informed pathway analysis. I have done this by developing comprehensive, high-throughput metabolite extraction techniques, enabling efficient quenching of the metabolome and recovery of the broadest range of metabolite classes. In addition, I have been developing LC-MS protocols for the simultaneous analysis of hydrophilic and hydrophobic metabolites in untargeted metabolomics. For targeted metabolomics, I've developed MRM strategies for quantitative analysis of chemically similar metabolites without the need for derivatization. I have also designed and implemented automated workflows to expedite data upload and metabolite identification, which I will discuss in detail in the next slide. And finally, I've been developing approaches to understand the biological roles of metabolites using pathway analysis, isotopes, and the recent implementation of a multi-omic processing tool on XEMS Online, which allows one to correlate their untargeted metabolomic data with other big data. As mentioned on the previous slide, I've developed approaches to improve the metabolomic workflow. Both data streaming and autonomous metabolomics have aided in speeding up the workflow immensely, saving weeks of mass spectrometry time and valuable sample. Thus, these technologies can have a direct effect on any researcher carrying out untargeted metabolomics. In brief, the streaming approach published in Nature Biotech has aided in reducing the mean wait time for metabolomic data processing by 7-fold. Instead of waiting for each mass spectral file to be uploaded and processed on XEMS Online, we stream the data after it's acquired and processing starts. Therefore, data upload occurs in parallel to data acquisition. Secondly, by using the power of the Metlin database, which has over 14,000 high-res MS2 spectra, we developed a method that simultaneously acquires both MS1 and MS2 data over the duration of a single LC-MS run. During the workflow, MS1 data are pre-processed by XEMS, features are extracted, realigned for retention time correction, and undergo statistical analysis. The MS2 data are acquired simultaneously using data-dependent acquisition, and the spectra match those housed on the Metlin database. Therefore, it's possible to acquire both quantitative and qualitative information in one metabolomic analysis and get instant validated metabolite identification. Finally, I would like to demonstrate how the impact of my research enabled the discovery of a novel metabolite and potential mechanisms associated with bacterial biofilms in human colon cancer. This was recently published in Cell Metabolism. Using an untargeted metabolomic workflow with targeted MRM, we identified and validated the association of polyamine metabolites with colon cancer tissues. Stratification by biofilm status, as determined by SEM, FISH, and sequencing, showed that diacetylspermine was upregulated primarily in biofilm-associated tissue and was confirmed by mass spectral imaging. Network modeling using the KEGG and Biosec database and pathway analysis using untargeted stable isotope-assisted metabolomics showed that diacetylspermine was an end product of polyamine metabolism. Using orthogonal biological techniques, it was revealed that diacetylspermine was produced by the biofilms, and biofilm presence induced a pro-carcinogenic state in the colon epithelium, further perpetuating the tumour. So the impact of my research has directly affected more than 11,000 researchers worldwide throughout my involvement in XMS Online, and has given the bioanalytical community powerful novel ways to identify and assess the roles of metabolites in pathways. In addition, I have published over 35 papers, and via the XCMS Institute, I have run workshops and provided online tutorials to bring my research findings to the public. Winning this award will be a great way to kick off my new lab at Yale School of Public Health, highlighting the impact of my work in the field of bioanalysis. Finally, I would like to thank my former advisors, Gary Schustak, Frank Gonzalez, Jeremy Nicholson, Ian Wilson, and John Linden for their mentorship over the years. Their support and guidance has allowed me to achieve my goals of having my own research program in this exciting field. Thank you for watching my presentation, and please vote for me at bioanalysiszone.com.